Hi, Fred. I just wanted to give you some idea of what we're up to and give you a little bit of the science that we're up to as well as we go through here. I'm sitting in a lobby area, so just across the street from Van Andel, where we were just looking at some cells that we're actually characterizing. So, um, what I'm going to do now is basically just run through some of the ideas, the strategies that we have for the lab, and then what data that we're going to be generating from it. So, and just to, I think, just give you an idea of where this support is actually going. So, one thing I just wanted to start off with was show you this sort of overall graph of, or graphic rather, that I'm seeing for how this fits in. So, I'm calling us this translational neuroscience lab because that's essentially what we've evolved into being. So taking together some of the developmental neuroscience that I've been involved in before and understanding how stem cells work, and then connecting that with biochemistry, and now being able to take that to looking at some models in terms of neurodegeneration, we're really being able to see how neuroscience can really work to help support um, uh, and understand how the nervous system works, and then be able to apply it to different situations. So here we are kind of sitting here in the middle. And then these different types of collaborations help illustrate some of the work that we're doing. So neurogenesis is being supported through and being executed through U of M, and currently is being supported by the Advance grant. And then we have the neuroprotection, which is basically trying to protect dopamine neurons, as opposed to here where we're trying to make dopamine neurons. I'll talk a little bit more about how that's actually therapeutically relevant. And then up here we have sort of how does NATO3 actually work, and it kind of overlaps both of these different things. How does this gene that we're interested in, that seems to be so important for helping to drive some of these dopamine making type neuron making genes, we want to understand how that works. What I've also drawn in the bottom petals here are some areas of potential growth, and just there's, I've heard about some types of collaborative types of funding and support that could come from Spectrum, so I'm interested in that and trying to pursue how that could work. That funding mechanism, as I understand it, basically relies on collaboration between different institutions. So I spoke to one of the people who does the reviews for that, and he was wondering, why we weren't in that. So I'm going to follow up on that. That opportunity is closed for this year at least. But and then also I think in terms of looking at what the MSU's College of Human Medicine offers, there's a host of people who are working and affiliated with the different labs there that I think are really good at being able to give us support, potentially even longer term hires in some way, to be able to help with some of the efforts that we have, be able to support biomedical science or chemistry or other types of departments that may have overlap with what we're doing here. So there's examples of two folks, one adjunct faculty in BMS and another one who's actually in chemistry, who have come from MSU and also Van Andel in terms of these kinds of postdocs. So that's something I'm interested in trying to develop more. It's really kind of fluffy at this point, but I just to try to capture it. That's what I did in this graphic I made for myself. And then here, what I have is this postdoctoral position is the one that it's a tech transfer type of network that is that the postdoc position is supported from. Right now we have Dolly Chowdhury, who's involved in that, and she's been a huge help in terms of actually supporting all three of these different types of efforts here, as well as just supporting and developing the lab culture. And so, with that petal aside, let's kind of dig in a little bit into what we have. So basically the gene, in terms of the translation of neuroscience, what we're thinking about is how how do genes work and influence stem cell behavior? And this is sort of the genesis of our work and where it came from. And we analyze how this happens using two different model systems. One of them is actually sort of hijacking or hacking into a developing embryo and looking at how their nervous system is actually developed, and basically overexpressing a particular gene of interest. So we shoot in a little bit of DNA, that's shown with this little arrow here, into the developing neural tube, and then from there, what we do is we allow the embryo to develop, and the green here is indicating areas in which we're actually having genes or being expressed. Then we do analysis with anatomical or quantitative PCR analysis or other types of strategies to be able to just determine whether there's an uptick or downtick. How do these cells change in response to the particular gene that we've overexpressed? And we do this also in cells as well, so this is something that's stemmed from the work that I was doing and kicked off in my sabbatical. So where we can basically reprogram cells or analyze and query questions using cells themselves. And there's different advantages to these different model systems that allow us to be able to capture the scope of the potential of a particular gene, or being able to understand some more of the biochemical mechanics behind it. So in its relationship to Parkinson's disease, basically what we have are different stages of Parkinson's disease, moving from the preclinical to the clinical phase of it. And the thing that's kind of useful in this kind of graphic is just looking at this is a relative amount of dopamine neurons here. As you lose dopamine neurons over time, what's happening is that you start to see different types of dysfunctions arise as you move along. And so initially there's this premotor stage. It's olfactory dysfunction, gut issues, things like that come up. And then finally, we start to see motor symptoms starting to arise. And then we also see this, the clinical ratings start to arise here. So that's when it's really kind of detectable. So there's a number of different strategies. So for example, Sukin Ku is working on trying to be able to work on biomarkers that'll be helpful for being able to capture this pre-symptomatic stage here. And then, and our work is basically kind of flanks and, or rather stretches across both of these. So we're going to talk about neuroprotection at one point. We're going to talk about cell replacement as well. And so the idea is basically, can we protect the neurons that are here and prevent this and change the slope of this line over time so that people won't suffer the ravages of the disease as readily? Or is there a cell replacement that we can actually do in which we're actually being able to add cells back that were taken out during the course of the Parkinson's pathogenesis? So when we think about how to do this, let's look at just basically trying to make new dopamine neurons and some of the factors that are involved in it. And so this list here shown in green and gray here are just different genes that if you look at what the genes are that are important for making and driving dopamine neurons, these are kind of listed here. And the reason that I did this and I kind of put this in this kind of graphic is that these types of genes and combinations of these different genes, along with multiple different factors, have been added to cells to try to help make them become dopamine neurons. And then those neurons have been then injected into animal models or preclinical models mimicking Parkinson's disease to see if it could help. And some of these strategies have actually been used to actually help without not the genetic component necessarily, but some other ones have been used to be able to try to help and generate these dopamine neurons in phase 1 clinical trials that are currently underway. So great, that's wonderful. There's, in addition, actually in Japan, there's phase 1 clinical trials that are using this kind of gene-mediated approach as well. There's also the possibility of potentially reprogramming resident cells within the brain using these different types of genetic factors. And so that's much earlier in this time frame. The proof of concepts just come out in the past month or two, where they've been able to take together some of these factors, including LMX1A, and SOX2 is another factor that's not listed here. And those have been used to help reprogram cells to become dopaminergic. And that's one study. It's in mice. It just came out, so it'll need to be replicated, but that's what we've got. Interestingly, though, this gene that we've been working on that we've modified is called phosphomimetic NATO3. We can see that it actually drives a whole host of these different genes that are known to be important in driving dopamine neurogenesis. And so we want to basically try to, the principle is basically, well, maybe we can add one gene instead of many genes and then be able to make embryonic stem cells become dopamine neurons really easily. And so there's some tricks to that, but that's kind of the idea that's behind it. So what I want to do is step back a bit and just kind of give you an idea of that one of the top petals that we had in there. So let's go back here. And if we look, so I talked about neurogenesis, I talked about neuroprotection, and I talked about this PMNATO3 and that it's driving genes that are important for neurogenesis. Those same genes are important for neuroprotection. And what I want to kind of draw our attention to, though, is here in this MOA, or mechanism of action here, because that's one of the first things that we've been kind of working on and kind of got us interested in all of this. So what we look at basically is we want to find out this one gene. What started it all was trying to characterize where it had actually been born. So this is an early stage chick embryo. So it's got the arrow here is indicating, if we looked at this bubble, if you will, here is just above the midbrain in the embryo. If you peeled that off, you would be able to see sort of a floor of cells there, and right along the midline of that floor, running all the way down, even down to the spinal cord, there's a stripe of neurons. These are sort of like, think of a keel of a boat, and that kind of thing. That's the midline in which we actually have this gene NATO3 that's being expressed. And so one thing that we were doing was characterizing how it worked. If we overexpress this gene, does it make the cells that reside within the midbrain? And this is interesting because it's from that domain, that sort of midline area, especially right up in here, from where these dopamine neurons that are born. And so perhaps understanding those mechanisms of neurogenesis there can help us be able to get insights into therapeutic mechanisms down the line. So looking at this MOA, the mechanism of action of NATO3, then we have a Campbell Foundation grant that's going to be wrapping up soon, but we'll ultimately be applying for an R15 to help kind of follow up in some of our work. But the question essentially is, how does endogenous NATO3 really work in the cell? We have a couple of formal hypotheses, but basically the idea is that that there's phosphorylation that's important for this NATO3 to work. So I'm going to step back just a second, actually, and talk to you about something. And that is, when we overexpress NATO3 in the developing nervous system, we find that we can only make new dopamine neurons here in this area. If we go a little bit further up here and overexpress NATO3 up here, we don't get dopamine neuron markers. If we go back here, we don't get dopamine neuron markers. And so that's for the unmodified sort of endogenous NATO3 gene. And so what we thought was, well, maybe there's something special about this particular area. And maybe the NATO3 protein that's expressed here is modified in some way, perhaps by phosphorylation, which is a common, what they call post-translational modification that can happen. So maybe that's what's actually driving this kind of effect. So what we've been curious about and what we've been working on for an embarrassingly long period of time is trying to figure out, does NATO3 actually get phosphorylated? So here's NATO3. We had a construct that we had to make, and that took a long time to do. But Dane Martinez, who's in the lab, along with Melina Frantzakakis, have been able to actually generate these epitope-tagged versions of NATO3. And it's basically a version of NATO3 that we can detect with they add an epitope on it so we can detect it by antibodies really easily. And so this kind of picture, the square here, showing two bands here and here. And then these are bands that represent size of a ladder. And the main idea that we want to basically kind of draw from this when we're looking at it is that this band actually represents the presence of NATO3 at the correct size. And so we can see what we did is we basically have taken cells that we grew, and then when we added NATO3 into it, we took those cells, we ground them up, and then we ran it out on this gel, and then we can identify using an antibody where our NATO3 is by its size, and so there it is. So what's interesting is that if you phosphorylate this protein with what's known as a kinase, then what can happen is that can change how this actually travels down the gel. It kind of travels down the gel in this downward fashion following its size and its electrical gradient. And so it actually is supposed to have an upward shift. And it's subtle here, but you can see that this is actually running at a slightly slower speed than this one here. And so what that's indicating basically is that there's some kind of phosphorylation event that could be happening that could be affecting how affecting NATO3, and that helps to support the idea that maybe this kind of phosphorylation could happen inside the body, could happen inside of the cells here, and that could be relevant for making NATO3 help to generate dopamine neurons here, whereas it doesn't in these other areas. So what we did then is that we took that NATO3 and that kinase, it was labeled PKA in the previous slide as C-alpha here. And the main idea is basically that we want to say, like, if we look at the first two, empty vector and NATO3 alone, it's a little subtle here because we have this large effect that happens down the line. But here's the open boxes are basically representing looking at these specialized midbrain cells that I obtained during my sabbatical. And what we're finding is that they actually, and this is kind of the baseline. It's like a negative control condition. And so what we're seeing there is that FOXA2, LMX1A here, and GRAILD1 and NER1 here, these are all they're empty vectors here, or they're all set at about 1, meaning that's, the rest of them are normalized to that particular level. And so what this is trying to say is that with NATO3 alone, we don't really have a change here of FOXA2, which is one gene that's important for making dopamine neurons. But we do see an uptick in this LMX1A gene, which is important for making dopamine neurons, and in GRAILD1, which is another one that's important for making dopamine neurons. We see this is actually quite subtle. It's like 1.6-fold or so. So it's quite small there, but this has actually gone up a little bit, which is consistent with what we've seen before. So the main idea is like, oh, we're actually driving up the expression of these dopaminergic genes, the ones that can make dopamine neurons or help protect dopamine neurons for that matter. So, but when we add this kinase into the mix of this, what we find is that there's a huge uptick in these genes, LMX1A and GRAILD1 and NER1 here. So they've gone up dramatically in response to that. And so what this suggests then is that maybe this phosphorylation with NATO3 actually is able to help drive dopamine neurogenesis, or at least the expression of these different types of genes. So where would this happen? And so this is actually data that we had a while back. So I'm telling you the story that makes the most logical sense, at least I hope, but what we're hearing. But some of the data we did actually, we got sort of out of sequence. But what this is depicting is DNA and the two bands of DNA here, and then these helices that are used to that represent, they're called alpha helices, and they're two different proteins that tend to dimerize and bind to DNA. And so this representation is showing NATO3 in blue, and then its binding partner is an E protein, is what they call it, shown in green. And they're not showing the whole protein, but just a segment of it. And basically what we did is that we went through and tried to predict different regions that would be phosphorylated by this kinase that I was telling you before. So this serine right here is, if we looked at, this is kind of giving letters that correspond to the amino acids that make up a protein. So this whole alpha helix is made up of all sorts of little, of these letters, if you will, just giving the building blocks of the amino acids that are part of it. And then we have serine, which is present here. And so serine is one that actually can be phosphorylated or modified in the way that we were talking about. And so that's one that we wanted to modify because it was actually a good candidate. It's one that if we look at proteins that look like NATO3, it seems to be an important one in dictating the behavior of it. And also it's consistent with it being a substrate for this kind of, this particular type of enzyme, this PKA or protein kinase A enzyme. So, okay, cool. So then what happened? Well, why do we care? One of the things that we can do is that we can actually modify this gene, or this and this serine here, for example, and then in turn use that to help and just test and see whether it actually does anything to drive genes that are important in dopamine neurogenesis. And so using that same cell line I was telling you about before, what we can see here is some of these genes like LMX1A, sonic hedgehog, and NER1, for example, the wild type NATO3 causes an uptick like here in LMX1A or NER1. And that's fine. Here's our empty vector. And but when we look at phosphomimetic NATO3, this kicks up LMX1A 20-fold here, NER1 10-fold here. So it's actually induced nicely the expression of this particular genes here. So that's been really good, right? We can actually see this phosphomimetic NATO3 that we call PM NATO3 is how it's referred to in this particular slide, is important for basically being able to help derive these different types of genes. And this looks a lot like what we see over here, where LMX1A is upticked here, NER1 went up, and this is all in the presence of that phosphorylation event. So excitingly, if we look over here at FOXA2 and NER1 within the developing embryo, we also find that this phosphomimetic NATO3 that we've made is able to drive the expression of these genes that are important for dopamine neurogenesis. And this happens broadly. If you remember, I was saying we can only make dopamine neurons arise in this particular area here. But with this modification, it seems to have made it so that we can make dopamine neurons all the way, moving all the way up here, and that we can make really interesting types of genes, not all, but some of the genes that are important for dopamine neurogenesis, even here in the spinal cord. So it's a profound type of impact that we're seeing here. And just to give you an idea of what this particular what this kind of slide is trying to represent, what we've done is we've basically gone through and cut and sectioned through this embryo, and now we're looking at it. And there's like a V here, which is showing sort of the lower part of this floor area. And the speckles here that are being shown are the speckles of, these are individual cells. And when they're lit up as white, what that means is that they basically are expressing our gene of interest. Well, on one side of the brain, we've treated it with our phosphomimetic NATO3, and on the other side, we didn't. And so there's a profound difference between the two conditions. So wherever you see white, that means we're expressing this dopaminergic type of gene, FOXA2, and then same kind of story as what we see over here. In this case, we have some of the tissue, surrounding tissue, non-neuronal tissue is kind of shown out here. So it looks a little different than what we have in this picture here, but you can see how we have these different speckles here and the expression of this marker for dopamine neurons called NER1. Lots of it over here, not as much over here. So, okay. This is really interesting because what this means then is that this modification that we made, where we took one of the serine here, we made it, modified it to an aspartate, then what we're able to do is make it so that we actually are mimicking phosphorylation through sort of a complicated series of assumptions. But it's almost like we've been able to take the gene and put it into the permanently on state, is the idea to think about it. So that we don't need to have the kinase around to cause this event to happen. Instead, we can just add our gene of interest. And so what this helps to do is kind of illustrate how the mechanism of action for that NATO3 in terms of driving some of these dopaminergic genes relies on what we think is that this kinase is driving it, and we can see that in some of our results, and then that the modification that we've made in it indicates that those regions that we've modified are important for driving that. And this is another marker that's, and the reason I kind of included this is because this one called Ingrailed1 is really important in terms of a lot of studies for, this is a gene that's not only important for making dopamine neurons, but protecting them. So here in green is, this is kind of a slice of tissue here. It's a little hard to see, but all along here, this is the left side, this is the right side, this is right along the midline. And then this picture is basically showing three different types of ways of looking at the same piece of tissue. Here we're just looking at the expression of a gene called Ingrailed1, and the spots here representing cells, and the green is representing where we've actually expressed our gene of interest, and then this is the overlay of the two. And so normally there's a little bit of Ingrailed1 that's expressed in this domain, and so we can see some of that over here, for example. But then there's other areas here shown with the long skinny arrows that indicate that these are that this domain is that there is that there's colocalization. Basically, we've been able to expand and increase the number of cells that are expressing Ingrailed1. So really interesting types of work here. So this led to the collaborations that we have going on now. So between U of M and the Human Embryonic Stem Cell Core, basically what we've been doing is trying to convert human embryonic stem cells into dopamine neurons. And so there's a picture of my collaborator there and some of the cells that we're working with. And then what we did is we basically made, this looks kind of like a Batman, so we thought that was pretty funny. But what this is basically human embryonic stem cells that are expressing our gene of interest. And in this case, the phosphomimetic NATO3. And so what we did is we actually made it inducible so that we can turn it on or turn it off depending on whether we add this chemical called doxycycline, which is simplified as DOX. So basically we just plate the cells, these human embryonic stem cells that we've made so it can express our PM NATO3. And the question is, if we add NATO3 and turn it on, this PM NATO3, this modified version of NATO3, maybe we can make dopamine neurons if we turn it on. So we add it here, and then we remove it at this stage, and then we let them develop. And so, and these illustrate the constructs that we used to do it, and that we actually upregulated the phosphomimetic NATO3, and also illustrate some of the constructs that we're using to that we've been making on our most recent grant. But here's the takeaway that we have so far in our data. So what we're looking at here is that this is a timeline, and then if we add our inducible, or this gray area here is basically illustrating when we actually turn on the expression of PM NATO3. So that's what's being shown right along it here. And then what we do is we shut it off, and then we allow cells to differentiate. So that's what's going on in this stage. So Nurr1 is one of those markers that's important for making dopamine neurons, and we can see that it goes up here and increases up to like 40-fold here. Whereas if we don't have Nurr1, but we just have some of the other genes that we were using to try to help drive those human embryonic stem cells to become neurons, then sort of our negative control, if you will, then they correspond to that blue line. So, so that's what we're seeing is that there's like about a 3- or 4-fold increase in this marker known as Nurr1 here. If we look at tyrosine hydroxylase, what's really interesting is that we see this huge uptick in induction of expression of tyrosine hydroxylase when we have the NATO3 present, the PM NATO3 present, versus what we have here in the blue line here. So, so that's pretty cool, because what that seems to indicate then is that we're, that we're able to drive some of these markers that are important for dopamine neurons. But it's not complete. We don't have quite the full story. Some of the other markers that we'd expect to be indicating that they're bona fide dopamine neurons aren't there. So even though we've turned on PM NATO3 during this period here, we aren't quite getting the full complement of neurons that we'd expect. So we need to keep playing with them. And so what we did is that we basically have made, and we're working on culture conditions that people have been using typically, and this is one example, where we've been able to take human embryonic stem cells. These are just different pictures of different human embryonic stem cell colonies, and then stain them for markers for dopamine neurons. And so tyrosine hydroxylase is one of them, and it's shown in green here. So within this sort of swath of cells here, we have the expression of, so there's this brightness of green here indicating tyrosine hydroxylase being expressed. And that's one of the bona fide markers for dopamine neurons. We need to see other markers come on, like FOXA2, which is expressed here. So that part's helpful in terms of being able to indicate that we actually have this expression of these different markers. And so there's some other markers that are being shown here to try to help rule it out. That it's not like a stem cell or something else. So it looks like this culturing protocol that we have seems like it works. And so what we're going to basically try to do is turn on NATO3, that phosphomimetic NATO3, and then add some of these different elements of culture and see if we can actually be able to really make bona fide dopamine neurons more quickly or more efficiently. Because this culturing technique, here it's 28 days, it takes a long time, and the reagents that we're adding are actually quite expensive to do to add to that. So this PM NATO3 might be really helpful in terms of being able to make that. And this is essentially kind of another graph that's showing the expression of our NATO3 gene over time, and then it goes up over, during differentiation. Um, okay, so that talks a little bit about how that previous one talks about how we make dopamine neurons and how we could make dopamine neurons. And the other project that we have going on is looking to see whether we can actually protect dopamine neurons using this PM803. So here is one where we basically have, we've got our gene, PM803, and then we're adding it into cultured human dopamine neurons. These are called Loomis cells. I'll show you a picture later on. And basically the idea of it is that we want to challenge those cells with either something that mimics Parkinson's disease through oxidative stress with a drug known as MPP, or another one that's important in terms of stressing the cells and forming aggregates inside of the neurons known as alpha-synuclein. And so this is a collaboration that we have with Van Andel. And so the same kind of set of enzymes here, or genes, that PM803 is able to drive are important for neuroprotection as they are for for neurogenesis. So it's kind of cool that we can actually take the same tool that we have here, that we've been exploring for a while, and then be able to apply it in two different contexts that both seem to bookend the potential to help people with Parkinson's disease. And so basically this is a timeline just showing some of the steps that we're at. So the postdoc that we have through support with T3N is now in the midst of making these lentiviruses and these cells here and generating them. I'm going to show you just briefly, a couple of slides here that have more data than what would be helpful. But what we're going to see basically is that we had to kind of choose some of the expression systems that we needed to be able to work and introduce our gene of interest into this human dopamine neurons, and then be able to put them into vectors that we want. And then we're working on generating lentivirus, which is a way of actually introducing that gene of interest, and put them into our cells of interest, these human dopamine neurons here. And then what we're going to do is test the effects on oxidative stress and cell survival, and a couple of other factors here. So where we are is that we're in the midst of making these vectors here. There's been some challenges as we've gone through there, but luckily we've had Dolly to help us out with that. It kind of goes through some of these different steps, and this just illustrates one of the vectors that we're using to try to help characterize it. And these little bands are indicating that we actually have the gene, at least with the insert present. We'll take those genes and we'll put them into, we'll introduce them into Loomis cells. We'll basically take those genes, we'll package them using the cell line here, and then take the virus that this actually makes and put it onto these Loomis cells, which I'll show you here. So just today I snapped this picture because we just thawed these cells out. We're going to be setting up those experiments soon. But these cells here, as we look at it in the dish, have the capacity to differentiate into dopamine neurons. If you add a drug, this has an inducible type of promoter in it. If you don't add the drug, then they'll just develop or continue to divide. So we're working with that and trying to see what's going on with it. So that was, perhaps maybe that was a bit long. So I always think it's going to be briefer than I thought. But what I wanted to do was basically give you, a bit of a, just like, let's, get into those details that are kind of, that are going on and that you might, that are happening in the lab and what has us so excited. And so what we've talked about, to kind of recap a bit, is that we've talked about how, we made phosphomimetic NATO3 because we were interested in this mechanism of action of this particular gene and trying to understand how it worked. And when we did, we found out that it drove the expression of all of these dopamine-related genes, making dopamine neurons. And so the next step then was to try to say, like, well, what happens? Can we protect dopamine neurons, or can we make new dopamine neurons? So those represent the two different types of grants. And so what the factor, and one of the things that comes in then is just, how do we see things moving ahead in the future? What is it that's going on? So currently we're supported by the Advance grant. So that's had a matching from Grand Valley, from both you and from the university, being able to help support the neurogenesis that we have here. And then we also have the NIH R21 that we've applied for, successfully obtained for helping work on this sort of leg of things. And then we have this mechanism of action. So looking ahead, just to kind of give you an overview, what we're going to basically be doing is that this has the commercialization potential to it that's most closely linked to the intellectual protection that we have right now, or intellectual property that we have right now. And so it's eligible for mechanisms of funding that we have that are linked to it. So I'll go through a list of some of those that we have coming up, but those have some match in it. The postdoc position also has match affiliated with it, and that helps to support all of these different petals of our work here, including this commercialization component. For this one, we're going to be applying for an R15 in February, and that one we'll be pursuing to try to see if we can actually get that to work. We'll also be applying for a Campbell grant to help supplement this R21 because there's an angle on it that's a unique one that we want to be able to pursue a little bit further, and hoping to be able to crack some issues that we've been having as we've been working with those Loomis cells that are showing you. So here's a list basically of some of the things that we have here. So that central petal, cellular mechanism of action for NATO3, the current Campbell grant basically is like $50K. That ends in July. And so we're going to be applying for an R15 of $300K. So that's going to be in February. What's probably going to happen is that we'll get some feedback. We'll have to reapply in September. But I'll be asking for a course release to match with that. And then then you can see this T3N support is kind of in all three of those, so I'll get back to that. Neurogenesis then here is what we have. That's I mentioned that commercialization part of it, so this wing of things. And so here we have it with the advances, what we have going on right now. We'll be applying for a Kickstarter grant and an MTRAC grant. These two have 4.2 and 10K matches. This is actually 10% of the total of the grant, so it's a much better sort of kind of a grant application than what we had with Advance, and so that's something that Linda Chamberlain was able to help argue for with the folks who are running this commercialization grants. What's really interesting is looking ahead, NIH has these STTR, sort of tech transfer types of grants that are used, which we can actually, are eligible, this is the indirects here, about 1 million that I've estimated. So 2021 and 22, that's when those would come up. So a lot of things have to line up for that. So that's hopefully that'll happen. But that's where things could potentially go. And then finally we have neuroprotection here, and so that's the R21 that we have going on now. So there's an R01 that we could have. I need to see, because if we apply for an R15, you can't have an R15 if you have an R01 already. So we have to see how these two things might coordinate in terms of when we apply for these things and how it works. But anyway, this data is coming along, but we probably won't be able to have sufficient data to apply in October of this coming year, but maybe a little bit later. And so we're going to use a Campbell Foundation to try to help fill some of the gaps there. And then the postdoctoral support. So this is one that in the past time that you guys have supported with Dolly, she's been an essential part of the lab here in terms of helping keeping things running. So that is renewable. That's at 50. The match is basically 25 or 50% of this the 50K, so 25,000 plus the benefits that are associated with it, and so that's 16K. So what this ends up being is basically the cost of the match and benefits totals 41K. So so that's what we're kind of looking at here in terms of match and sort of how it fits with some of our goals within the lab. I hope that was helpful in terms of trying to illustrate something. I want to also show you another thing. I'm trying to end this show here. I'm going to include this spreadsheet for you as well. But what this chart is trying to illustrate is a little bit about trying to look at the money side of things and how it links to plants. So I talked to you before, we have commercialization here, neuroprotection here, mechanism of action here. And so those are those three different petals in the top part of that flower that I was showing you in that one diagram. And so what we have right now is we have a T3N that's helping to support that. And that was the amount that that was being covered was 25K. And then we had to match it with 41K to come up with our total, basically, of 66 to cover that postdoc position. There weren't any indirects associated with that. Here's the advance. This is one that you guys also supported. So 25 that you put forward, a match of 25, and then so the grant total there was 50K. So so naturally, this is like you know, and what I'm showing along here is just a timeline, and basically, if it's in dark green, it's things that are funded currently as funded projects, and light green is just showing where we anticipate this timeline actually occurring, and when we're doing it. So I thought this was useful because it kind of gives you an idea that, you know, there's been some substantive types of investment so far, but as we look ahead, what are we really kind of dealing with in terms of you know, matches? Am I going to just kind of keep coming back more and more to you guys, or is there some kind of growth that's going to be happening here that's meaningful? And so for the Kickstarter grant that's coming up, this application's due in May, and so this match is 4.2 thousand. Our MTRAC here is about 10,000. So these both fit under that commercialization, turning human embryonic stem cells into dopamine neurons. So these two we should be able to apply for and be able to get the money for that. I would think. I think we're in good position to be able to do that. And so that kind of gives you the timeline for it. And then as we look ahead, then we're in the position to be able to apply for what's known as the STTR grants. And there's different phases to them with different amounts of it. So if we go in the standard path, basically it's 150K. I would ask for a course release to help manage that grant. And then there's, so the direct costs are 150, and then there's the indirect costs that I estimated from a 41% indirects. And then we're looking at what happens for the phase 2, and that's what we have here. So this is pretty healthy as far as a sum. We're looking at about 41,000. If we go to the phase 2B here, then we end up in the millions, which is really nice. So 1.2 as far as the indirects, and so $3 million grant to help support that. So this is a little harder to get into. So at this point, the stakes are higher in terms of how translatable is the technology, and how much have we been able to interest commercial partners in it. So I think these first two we're good with. I think we may be able to get through to phase 2 here, and then we're in sort of an area of having to really wonder where we are in terms of the quality of the data that we have so far, and what we're and you know, whether we're able to kind of interest those partners or not. So it's going to take some work, but hopefully we can get to that other phase, and then ultimately into venture capitalism. For neuroprotection, then what we're looking at is this. The R21 that we currently have, and then we'll be applying for an R01 as co-PIs. And this is contingent upon the data being good. So if we don't see anything, then we can't apply for the R01. So the R21 really does allow us to be able to test and see whether we actually protect dopamine neurons or not. And the experiments are good, so we should be able to know our answer, but hopefully we will be able to be in the position to actually apply for it. And then as we look ahead, this mechanism of action, what we started with at the beginning, if you remember me showing those blots and things like that, there's a couple of things here. We have the first Campbell that we have here, Campbell 2, this is the second Campbell that I'll be applying for. This is helping to support the neuroprotection, but also fits in here. And so that one has a match of 6.9. And then we have, it's a $50,000 grant. And then what we're looking at here is we'd be applying for an R15. And so I've been talking to one of the people who's actually on the committee for that R15. I'm excited about it because he was really enthusiastic about our university. So we're looking at basically trying to apply here. We may have to reapply. If not, then it'll start. It'll start at this point here. So it'll be pretty nice as we're looking into. You know, when it starts up in November. But otherwise it would be shifted downwards accordingly. So, and then the second round, you can renew these, and so hopefully we'll be in a strong position to actually be able to renew that R15 for the second series of 3 years. So that's what we're looking at in terms of how these match dollars work. The other, T3N, this T3N we're looking to actually apply here. And this is the one where we'd be looking for a match of 41,000 to help cover it. So I'm talking with Linda. We're trying to find out if what our sources of match are to see whether that's something that we can do to help keep the momentum going for what we have on this postdoc position that's helping to support so many of these different aspects of our work. But that's one element of it that's out there. So if we were to tally up the matches that could potentially come up in 2019, then this one is one of them at 41. Then we have the Kickstarter and the MTRAK that are coming at 14.25. And then the Campbell here at 6.9. So those are the ones that have a match component to it. I'll be applying for the R15, and with those NIH grants, I would be asking for a course release as a match, since to help make sure I can manage the grants effectively. So that's a little bit about what we're up to, a little bit about how the money part fits into it. I wrote this out as well, and but I thought I'd also spend just a little time, I guess 46 minutes and 30 seconds going through all of this. So if you listen to the whole thing, I'm proud of you. And thank you for your support, and we'll talk soon. All right, take care.